Hello and welcome to Biology 11. Okay, now in the Biology 10 I mentioned that I was actually going to speak about prokaryotic cells today. So that's exactly what I'm going to do. So yeah, all right, so let's make a brief note at the beginning though. We're going to be moving towards talking about prokaryotic cells. We're going to be moving towards talking about microscopes as well. So a good port of call here is to look at the units. So we've got the micrometer, and that's denoted by this is mu, which is a 12th letter of the Greek alphabet, mu. So we do it, and it will be micrometer like that, and that is equal to a millionth of a meter. Or yeah, or 0.001 millimeters. Yeah. So that's one way of looking at that. So yeah, so probably 0.01 millimeters. So it's basically, so if we multiply by 10 by 100, we get to 0.1. If we multiply by 1,000, we get to a millimeter. Yeah. So it's pretty small. So 1,000, yeah, 1,000 to get to a millimeter. If we multiplied that by 10, we get to a centimeter. If we multiplied that by 10, we get 10 centimeters. Multiplied by 10, we get to we'll just it would just carry on like that anyway. So a millionth of a meter, or 0.001 millimeters. Also, we look at even smaller things than that, which is a nanometer. So a nanometer it's going to be, let's have a look at that, 0.00, and then we've got another 3. So we could say this, nanometers. So that is going to be, a nanometer is going to be, so there's our million. So it's going to be 1,000 millionth of a meter is 0. So yeah, so that's going to be 0.0. It's going to be 0.0000001 millimeters. So a nanometer is the same as that. So we've got the micrometer and we've got the nanometer, and we can see that there's an order of three orders of magnitude here. Okay, so that's just a little bit note about the actual. This is like actual size. Okay, so we're looking, and yeah, this will be clear actually as I talk about the topic here. Okay, so yeah, so I've spoken before about this, that so prokaryotes. Okay, so prokaryotes, they're interesting. This carry section is interesting, but anyway, so prokaryotes always single-celled, always. Eukaryotes. Okay, so we could put this. So we could just do this. Put eukaryotes here. And then we're going to do a line here, and we'll just continue this line down. There's a few things to talk about here. So these eukaryotes can be single-celled, or multi-celled. So you can have both. But prokaryotes, there is a strict single-celled look to it, and that yeah. Okay, so listen, I'm not going to try to say what these things suggest because these are very complicated topics. So anyway, so we're going to move down. So this is going to be like yeah. I'm not. I could put do this in a list form, but I'm doing it in a list anyway, and I'll explain it along the way. Okay, so the DNA is circular. So you have like the. This is obviously not But yeah, yeah. This is funnily enough. This is actually a maths problem. This thing. So circular DNA and a eukaryote's DNA is linear. So I guess we could just, I don't know. Maybe that's not a good representation, but anyway, it's one way of looking at it. It's not. It's not. Yeah. Okay. So with prokaryotic cells, no nucleus. So there's a big thing. So recall that what we said about the nucleus. We said that it was it had the nuclear envelope, had chromatin, the nuclear pore, and it really is. It's like the control center, isn't it? Yeah. The chromatin is made from DNA, and DNA controls the cell's activities. And the pore is going to allow the ribonucleic acid to move between the nucleus and the cytoplasm. And the cytoplasm that we mentioned last time, it's got these microfilaments, microtubules, and is very much like a skeleton that strengthens it. Doesn't means that like it's like. Just imagine it like a spine. I mean, yeah. Imagine you can imagine it like it's a spine, or a very important part of the skeleton, which helps to maintain the shape, or like the the thorax or something like that. Okay, so yeah. I think that's okay to say that. So anyway, no nucleus, DNA free in the cytoplasm. Okay, DNA free in cytoplasm. Okay. Yeah, and we recall there's actually the nuclear. I probably did say it, but I remember in when I was doing the research for Biology 8, I talked about the nuclear plasm as well. Anyway, so nucleus present. So DNA is in the nucleus. Okay, that's pretty cool. Not the best. All right. One of the things I noticed when I actually render out the footage, sometimes it actually it's a bit harder to read. But I mean, I can still read it. It's not terribly hard to read, I suppose. Anyway. So yeah, so DNA is inside the nucleus, as we've established and I just mentioned. Okay, so let's carry on with this. We'll just draw this line a little bit further. It's getting interesting curvature to it now. Okay, so the cell wall in prokaryotic cells is made of a poly. So there's probably many, I guess. Saccharide. Well, the only thing I think of there is saccharin, I think, which is added to. It's like a sugar. It's like an additive, isn't it? A famous one of a polysaccharide called. What do we have here? Glycan. I think glycine may be an amino acid. Is glycine an amino acid? Like I said, I'm pretty. Oh no, no. I've got that completely wrong. Hang on. So yeah, I just need to. I don't think I actually am that far off the mark here, but I'm not sure. Yeah, the cell wall seems to be an interesting thing. It's the only reason I stop on this one because I ran, yeah. No, I mean, yeah, but because it's just that, yeah. So anyway, so that's what it is. Polysaccharide called peptidoglycan. And obviously, I'm going to have to try to actually learn how to pronounce the names. I was thinking of even using like there may even be I could use some sort of diphthong method or something like that. I mean, there's maybe how I have to do it. But anyway, I can't. That may not be the right word I used there. I think it is though. I think it is. I do tend to look at certain things. So anyway, so no cell. Yeah. So you recall there's no cell wall in. If you look at my pictures in previous episodes, you'll see there's no cell wall for animals. And yeah, there's a cellulose cell wall. Cellulose cell wall in plants. And you'll recall, actually, one of the things that strikes me there when I talk about that, I remember doing, I spoke about an interesting cell wall. Yeah, gelling. Yeah, I mean, there is a gelling agent. Yeah, lignin. Yeah, that's the one with in xylem. There's a containing lignin, strengthens the water principle. Yeah, that was the tree one, I think. Yeah, lignin is pretty cool though. I remember being quite interested in lignin when I looked that up. But anyway, so yeah, I'm just adding a few little touches here. I probably should just focus on the topic though. And then a chitin cell wall in fungi. Yeah. Okay, so that's obviously very important. So so there's no membrane-bound organelles. So it's almost like we're looking at to look at these. Yeah. Okay. No membrane, no membrane-bound organelles. Okay. So, for example, mitochondria. If you recall in Biology 8, had some nice pictures of the mitochondria with very nice membranes. But there's nothing like that in prokaryotic cells. Whereas in eukaryotic cells, of course, there's many membrane-bound organelles. And in some cases, when I did look at the internal structure, that's one thing I do need to do, look at these different words with internal structure. Because I looked at the word ultrastructure, didn't I? Whether an ultrastructure is a good term to use, it may well be though. Many membrane-bound organelles. Okay. Oh, I didn't actually include. Which one did I actually start on here? Always single cell DNA. Oh yeah, I didn't actually include the size. Okay, so let's just talk about that. Okay, yeah, this will work out quite nicely actually, because we can talk about the size in a separate little topic here. So yeah, so prokaryotic cells extremely small. These are very, very tiny little things. Extremely small. Less than 2 micrometers in diameter. Okay, so imagine the cell and the diameter going through the origin there is going to be less than 2. What is the best way to do this? There doesn't seem to be a good way to write that, but anyway. Alright, so yeah, and I spoke about the actual size. So if we're looking at 2 micrometers, a micrometer is 0.001 millimeters, so we're looking at less than 0.002 millimeters. So yeah. So and I'll talk about what kind of microscope could see that. It shouldn't be too. Yeah, you just need to think about doing those things, and I'll do that. But yeah, it's not. I mean, it's small, but it's not. But I mean, as a thousandth of a millimeter is small. But you can see it. You can see a millimeter. You could see 0.1 millimeters. So you only like 100 order of magnitude. You probably with 100 times microscope you could see the magnification. Okay, so yeah, eukaryotic cells are larger by comparison. Larger cells. So we're looking at between 2 to 200 micrometers there. So a little larger. Um, and yeah, so we're getting almost close to the millimeter scale, really. So they're not. I mean, so they're really. Um, yeah, possibly. I mean, yeah, I imagine you probably could see one of them. It probably wouldn't. You wouldn't be able to tell much about it. It'd probably just be almost invisible, but it's just beyond. Probably a different mammal could probably see it better. But obviously one with good eyesight. All right, so then we've got small ribosomes. And these ribosomes are 20 nanometers. So we're way off. We're not seeing these ribosomes anytime soon. And these ones are larger. So yeah, there's not too much. So yeah, so that's larger ribosomes. Ribosomes are pretty small though. So yeah. Okay. And just to get some clarification what these are, so we can have here E. coli, which is a bacterium, and Salmonella, it's a bacterium. Yeah, and of course the is singular, and bacteria, for instance, is plural. Okay, and then the eukaryotic cells often have a kind of more kind of is be like human liver cell, yeast, and of course amoeba. And I think much